And good afternoon, everybody. I'm pressed to see you all here at 5 o'clock in the afternoon after what has been, if you're like me, a pretty intense day already. But we don't intend to allow any napping because this is going to be a very vigorous panel with a lot of interesting materials to share with you, and hopefully we'll get a discussion going that can be pretty interactive amongst the panelists who have arrived here with all kinds of expertise and information to share with you. I'm Francis Collins, the director of the National Institutes of Health, and we have an entire institute called the National Institute of Aging that supports research that we're going to be talking about here in this 1-hour session. And my own laboratory actually works on a rare form of premature aging called progeria, which is my other little bit of insight into this. But the real expertise is the 4 people you see up here with me, and I'm going to try to convince them to share all kinds of exciting stories with you, but I don't think I'll have to try too hard. Just to kind of get this started a little bit, why don't we show a quick video of what maybe we would all hope someday to be able to aspire to. This is a video that I got from near Barzilai. So can we run that video for a moment? It's only about 2 minutes.
In this office building on Madison Avenue, New York City, 104-year-old investment advisor Irving Kahn is working hard, as he has since his career on Wall Street began in 1928. He shares his secret to a long and healthy life.
To wake up in the morning and have something to look forward to.
Irving's curiosity and keen business sense have led him to become a widely respected value investor, a member of the New York Stock Exchange, and chairman of Kahn Brothers, the company he founded more than 30 years ago with 2 of his sons, including Thomas, who is the president. Irving works 5 days a week with his 67-year-old son and 29-year-old grandson, Andrew.
And how are you going to link the underwriting projects?
How come that?
Playing an integral part in managing over $700 million in assets.
This is the Aztec website.
And Irving says not working is unthinkable.
Well, I would pay you if you took it away from me. I try to buy it back.
He believes mental challenge is key. To stay sharp, Irving reads materials online, 2 financial newspapers daily, and a wide range of nonfiction.
I read a lot of science. I read no fiction, no mystery stories, and no sex novels. So that leaves a lot of time for science.
And it was Irving's interest in science that led him to participate in the Longevity Genes Project at Albert Einstein College of Medicine, led by researcher near Barzilai. Irving and Thomas are part of the study, as is Irving's big sister,
Muskets is 5 feet long.
108-year-old Helen Reichert, a former television host and fashion historian. So far, Neer and his team have found several gene variants that are more common in this group and protect against cardiovascular disease, type 2 diabetes, and Alzheimer's. Neer's team has also found that some genes seem to protect against the effects of certain lifestyle habits.
You can see both from him and his sister that those genes have real amazing effect, not only on the fact that they're alive, but considering the fact that they were smoking and this should have shortened their lifespan immensely.
And Irving believes long life has its benefits when it comes to business.
Because part of it is some of the advantages that you have when you're older. And you know what's rotten and what's fresh and what's good and what's bad. And you can participate on a higher level.
How about that? So our panel this afternoon, over here on my far right, your left, near Barzilai, who you just saw in the video. He's the professor of medicine and genetics and director of the Institute of Aging Research at the Albert Einstein College of Medicine. Next to him, Nobel Laureate Elizabeth Blackburn, now president of the Salk Institute. To my left, Joon, who is the managing partner and president of Palo Alto's Investors. And then on the far left, your right, Cohen, who is the dean and the USC Leonard Davis School of Gerontology professor. So lots of expertise here. Maybe just to set the stage, let me say 3 quick things as far as principles that we're going to talk about, and then I'm going to pose questions to these folks to get this started. First of all, what we're really talking about here is aging as itself a risk factor for common illnesses like heart attack, like stroke, like diabetes, like cancer. But why do we study each of those diseases individually instead of studying the whole field of aging as that risk factor and see if we could reduce that? Basically, focus on how do we extend the health span of individuals. We talk about lifespan. Today we're going to talk about health span, the opportunity to live a life that is free of those kinds of illnesses which come along all too often as a consequence of aging. That's one thing. Secondly, we want to talk about the biology of aging. We're learning a lot about this. Maybe it's not just sort of a running down of the system. There's some stuff going on here that you can track. There are markers that can teach you something about biological age, which might not be the same as chronological age. We'll be talking about that. And then third, and probably most importantly for any of you who have started to wonder about your own aging process, we are getting into a space now where this is not just an academic conversation. We have potential abilities to target the biology of aging and do something about it, although it's early days to see exactly how that's going to play out, but we'll be talking about that as well. And we do know that in experimental animals, we're getting kind of good at that. We can make mice live substantially longer than their normal lifespan. Okay, we're into mammals already. How long will it take before we get to humans? So those are the things we're going to talk about. And without further ado, let me start the discussion off, and maybe I'll pose my first question to Liz Blackburn. So Liz, you have won a Nobel Prize for work that you've done, which has been one of the most fundamental observations about aging and the process that clock seems to run, and it relates to something called telomeres. So can you tell all of us what's that about, and where do we stand as far as our understanding of how that plays a role in the aging process?
Well, it all began with, really basically trying to understand something about our chromosomes carry all our genetic material, but the chromosomes in bundles of DNA, they have ends like the little protective caps at the ends of some of our shoelaces, and the shoelaces tips, if they wear down, they let that shoelace fray away. So it's a little bit like a telomere at each end of your chromosomes is a little bit like a protective shoelace tip. And they do wear down as we humans age. And so I was really just trying to understand what was at the ends of chromosomes. And then what happened was that we looked in model organisms, and yes, if you took this ability of the replenishment of telomeres to happen away, and we discovered an enzyme that can replenish telomeres. And if you took it away from mice, they eventually got old quicker. But they don't die of telomeres wearing down. But we live 40 times longer than a mouse, sometimes longer. So the question was, does it actually naturally wear down in us humans? And the answer was, oh my goodness, yes. And does that matter? And now cumulatively, there's very clear accumulated research from just all sorts of aspects of human genetics, human cell biology. We can say, yeah, this is a contributor to those multiple things that Francis pointed out, the cancers, the diabetes, the cardiovascular disease, the things, and even some dementias, the conditions we die of. So we really care about those because those are what limit our health span. So if we're going to go into a longer health span to get us out to the point where I always like to say our genes can do us some good, then we need to get past this set of big impediments to extending our potential, and that is, how do we minimize the chances of the big killers of humanity? Mm-hmm. And one of the things is to realize that is one of the things going on. If we can modulate that wearing down of the telomeres, that'll help us. It won't get us all the way, but it'll help us get lower chances of those things we don't want to get as our birthdays tick by. Other things go on as we age. Proteins in our brains get old. That's a different kind of aging aspect, and that's going to be a very exciting emerging still is emerging kind of science. There are other things that go on as we age. I've just named 2 of them, and I think we'll hear more from the panel here.
I'm sure we will, and
but we've got some
from you
power.
and others.
We have some power to do something about this wearing down of the telomeres, and a lot of it is in our hands right now, and it's a lot of lifestyle things that we knew already, but we can quantify their effectiveness by how well they prevent the wearing down of the telomeres. So that's one handle on our health span that at least we've got some good grasp on now.
Terrific. So you mentioned, of course, genetic factors that play a role, and we saw those siblings in that remarkable video who are well over 100 years old, and nobody has studied the genetics of aging more carefully than near Barzilai. So what have we learned? What if we got to pick our parents, and better yet, pick their genomes, what would we pick?
Right. So I just want to make sure that you know also that there are 2 other siblings. So there were 4 siblings that were born to 2 parents in New York between 1910 and 1920. Each one of them reached 102. Helen, the sister, died when she was 110. And she smoked for 95 years. First of all, so that's genetics, okay? But first of all, if you smoke for 95 years, I promise you live long life. But the point really is that she was resilient to the effect of smoking. So another important thing for me to realize from, by the way, I have 700 people like them and their families, their children, okay? So I saw a lot of those people, and the first question that was important to know is, did they get sick when everybody gets sick, and then we're sick for 30, 40 years and die? Or was their lifespan linked to their health span? And the answer is, and it's true in every centenarian study in the world, is that their health span has been extended by 20 and 30 years. So their longevity was associated with the health span. And so I'm maybe biased because I have those people who at 104, they are healthy, and that's why their life seems so good. So the other thing is that they die relatively rapidly. They have what we call a contraction of morbidity. They live, and then many of them die one day. The CDC has actually characterized what's the last 2 years, the cost, the medical cost in the last 2 years of life of somebody who dies at 100 or somebody who dies at 70. And it's third the cost to die at 100. And when they were 70, they even didn't go to the doctor because it wasn't available now. I want to stop now. I'll come to the genetics later.
So, but before you completely stop, have we actually identified specific genetic variants that are associated with longevity, or are we still looking?
No, we have identified SNPs that across population, SNPs are just genetic markers that are different than in the majority, okay, in important genes that are associated with longevity. But more important, in subpopulation, we discovered functional mutations that are consistent with what we know with aging and in genes that are related to aging. Hassi and me have actually found several genes that are really in the middle of the growth hormone pathway. And maybe in California you think that growth hormone is anti-aging, but actually it's the opposite. If your growth hormone is not functioning well, just a little bit, you're more at risk of having longer life. So this is an example of when we have several of those. This is an example, and we have more example of genes that are very important for longevity and more of women than of men.
Let me ask Kasi then to say something more about that. What do these genes do? What are we learning about what really is the basic biology of what goes on that determines whether somebody's going to have a long health span or not?
So it's interesting. Cells in different parts of the body age at different ways. And inside the cell, there are different components of the cells that are more sensitive to wear and tear over time. So the most famous, of course, that Elizabeth described already is the telomeres that get shorter with age. Another really important part of the cell is called the mitochondria, which is the energy source of the cell. And the mitochondria is, for some reason, much more prone to break down and stop functioning well with aging. And because it's a source of energy for the cell, and because it's so important for metabolism, many diseases of aging and the aging process itself is often associated with decreased mitochondrial function. So, for example, we discovered certain hormones that come out of the mitochondria, and we used Near's population to show that families of centenarians have much higher levels of these hormones that come out of the mitochondria that may support a healthier, more energetic aging process. So I think the key is that different people and different parts of your physiology age in different ways, and we might be able to personalize our approach to targeting them.
So this sounds like an area that potentially could lead to some pretty significant medical applications, not just basic science discussion. So Joon, let me ask you, you after all are managed partner and president of Palo Alto's Investors, LLC. So what is the climate here for investment in aging research, and where's it going?
Well, historically, there's been very little investment in aging. There's structural reasons, such as you can't run longevity trials. It takes too long. But when we think about what the opportunities are going forward, given all the emergent science that we're talking about, there are a couple of things we undertook. One is actually through philanthropy, so impact investing, and the other is through more traditional investing. In terms of impact investing, we launched through a prize called the Palo Alto Prize. And prizes are wonderful because it's a way to lead science in a new direction. So here are the areas that we're super interested in. The first principles of biology are maintain homeostasis and increase homeostatic capacity. So you can think about healthcare and the first principles of healthcare as restore homeostasis when somebody's about to die, and otherwise try to improve their homeostatic capacity. That's the way we're hacking our biology towards better health. Now, homeostatic capacity, you can think about it as your ability to get to homeostasis. Remember the toy, the Weeble? As you push it, it self-centers. That's essentially what nature endowed in us. And after you turn 40, or as we age, we start to see the decline of the ability to self-center, auto-regulate, buffer, coping mechanism, resilience, robustness. They all mean the same thing. So I turned 40, and all of a sudden, I couldn't ride roller coasters because my Weeble would wobble. I couldn't do somersaults. I couldn't recovery from almost anything took longer, whether it's an injury, a late night jet lag. My Weeble was wobbling. In fact, I went to a Lady Gaga concert on a Tuesday, and I was still tired on Saturday. I remember being able to stay up all night when I was young with impunity. My pupils don't open up in the dark, so I can't read menus at night. Our tolerance for thermal variation, altitude variation, it's all declining. These are the subjective things we can feel, but objectively, we're also losing our homeostatic capacity in very subtle ways. When we're young and our blood pressure or sugar goes high, our homeostatic capacity returns to homeostasis. Whereas now, after 40, in some of us, they go high and stay high, and we give it names like diabetes and hypertension. So a parsimonious way to understand all the epiphenomena of aging is it's lost homeostatic capacity. Maybe it's not inflammation per se, but it's lost inflammatory capacity. So if that's the case, the prize was launched with the premise that if we can improve homeostatic capacity, could we actually improve health dramatically to the point where you can avert aging? That was a premise. On the investment side, there are 3 areas of great interest for us. One of them is on biomarkers. So the biomarkers we're using in healthcare today are largely state variables. Now, the body's a dynamic system, and using static variables to assess dynamic systems is very hard. To give you an analogy, when we go to our annual checkup right now, we measure your heart rate, blood pressure, glucose, and cholesterol. Well, that's like a seismic scientist trying to forecast collapse risk of building, checking on you once a year based on how much it's leaning. Now, what they actually do is they do something called resilience testing. So we're the only field of science that still is stuck in state variables. We should be thinking about dynamic variables on top of that, measure the resilience. So we are contributing IP, and we're investing in areas of essentially stress diagnostics. Remember, your heart rate doesn't really change much with age. It declines a little bit. But your heart rate response to exercise, what actually is declines with age, and it maps very well to disease of aging, and to mortality rates, and not dying is a great way to promote longevity. It's amazing that we don't use it more often. And then on the therapeutic side, rather than just trying to improve your homeostasis, what if we try to improve homeostatic capacity? So interestingly, the best way to treat high blood pressure is not to lower it with an antihypertensive, is to, quote, raise it with exercise. The more times you raise it, the more your homeostatic capacity improves, and this actually restores health. So what if we gave people blood pressure raising medicines as a way to treat the high blood pressure? That's okay, it's the Hippocratic Oath. We never really considered this. So in the investment side, we want to focus on things like that where improve homeostatic capacity as a way to return health. And then the last area of great interest is what we heard around the table, which is around the genetic programming. Ultimately, it seems like there's genetic and epigenetic programs that affect aging, and there's enormous plasticity to it. Are there technologies we can invest in to really improve our health span that way? Those are the 3 areas of interest.
That's great. So I'm sure we're going to come back to in a little bit here, what does it take to actually begin to initiate something like human clinical trials where, as you say, it takes a long time to prove that you've extended somebody's health span, and we probably don't want to wait that long, and so we'll maybe talk again about biomarkers. But let's talk about a couple of the interventions that are looking interesting in animal models that if we got to that point, we might want to try. One that was just published a couple of weeks ago, and I'm sure everybody read my blog about it, right? Because I blog every Tuesday about something that has come up that's exciting in the scientific literature, and this was the paper I chose last week. So this was a follow-on to a previous study which had looked at what happens if you put mice in a situation where you have an old mouse and a young mouse, and you exchange their circulations. They're sort of zipped up together so that they're sharing the same blood circulation. And what happens is you slow down the process of deterioration of the old mouse. There's something in the young mouse blood that seems to help. Sounds very vampire-like, but and it's created all kinds of bad cartoons and jokes. And we'll come to Nir in a minute because he actually runs a lab that does this kind of study. What was published, though, 2 weeks ago was an application that kind of took my breath away, where they decided, well, let's see what we could do if we didn't just do mouse to mouse. What if we did human to mouse? And let's try to pick the human source of blood plasma that would be as young as possible. What would that be? Umbilical cord blood. So they took human umbilical cord blood, they spun it down, they took the plasma, they infused that into an old mouse, and what do you know? The mouse's hippocampus, which is responsible for memory and other things, got a lot more impressive, both in terms of its anatomy and in terms of the mouse's function. And that didn't happen if they did this with plasma from an old human into an old mouse. And they even claimed by going through some experiments that they understood what component it was of that plasma, something called TIMP2, which is a metalloprotease, which is not the one I would have guessed. So I read that and I thought, somebody out there is probably starting a business on this, which is probably not going to be approved by anybody, where you have the opportunity, I hope none of you have done this already, to get a little umbilical cord plasma zipped into your system when you're sort of forgetting where your keys are. I sincerely hope we're not at that point. But Nir, what's going on in this field? Is this like one of the biggest clues we've had in a long time, or is it still like, well, maybe, maybe not?
First of all, my knowledge comes mainly from your blogs, so you've just described.
I knew I picked the right people on this panel.
But it really started, you know, he woke up this morning with $2 billion more. I have to be nice to him. I wore a pink shirt and not a blue shirt. But it really all started with this very interesting paradigm, scientific paradigm, where you take two mice that are genetically similar in almost everything except maybe one thing, and you tie them on the side, you tie their skins together so they share blood supply. Now, you can do it between young and young animal or old and old, but the interesting thing is to do it between young and old. So you put them together, and by the way, it takes a while to know how to do the surgery well, but the young and young look like Siamese twins, and the young and old are kind of becoming pals to each other. One is more of a bully, sometimes it's the young, sometimes it's the old, but it's kind of a nice experiment that is not stressful. But one thing happens, the old becomes young, although in certain ways the young become old. And thinking of this paradigm, there's a lot of what we can do in order to identify those plasma factors that can affect aging, and that's what led to young blood in old animals and to this experiment that you're talking to, and to the commercialization of these things.
Yeah, potentially.
But I'd like to add to that to say that I think it's a great biological experiment, but it's all about discovering that particular molecule, TIM2, for example, which I'm sure there are many others that contribute in other situations. Yeah. And we really, take that embryo out of your pocket, please. But we really don't want people to be injecting cord blood. What we want to find are the substances that help you, that are found in cord blood that can help you age more slower.
Correct. Indeed.
And also, maybe you can get your own cells to be rejuvenated. So another mouse experiment that I loved came from the Salk Institute, where they put some factors in. These were things that you can introduce by, like a little harmless virus that doesn't even make you get a cold. You can put these factors into cells, just introduce them, and the cells get partly younger. They don't go all the way back to really young, but they get younger and they start to be able to replenish the tissues better than in the old mouse. So the mouse really reversed some of its aging. Now the real question is, what will be the thing that will play out in our long human lives? And I don't think we'll have to wait all that long, because I think there are a lot of ways we can take biomarkers and say, okay, we're going in the right direction here. So I suspect there'll be results that will start to be reliable before waiting for 80 plus years or something, or 100 years for your population. I bet we'll have some really good guideposts as to what things we can start intervening with humans. And I think, it's a great thing to think. You asked about biology, right?
Yes.
So let's think about what lasts and never gets old, and that's our germline, because, generation after generation. So we have cells in our bodies that know how to make a completely new baby each time, a young one. So those cells, they've learned how to stay, quote, young. So if we can learn their secrets, we might learn some very interesting things that we could start applying to, as Hassi was saying, the different kinds of tissues. Some age faster, some slower, but there's some tricks that our cells in our own very bodies have already figured out, and diving into figuring out those, and there's some clues coming from those. We might be able to really start intervening in ways that I think will take, careful thought and application, but this is not science fiction anymore. This is real biological understanding more and more as we look inside this.
In order to make it relevant for humans, we've got to get through, of course, clinical trials. And that's where we have an interesting challenge, because, again, I think the lifespan component in animals we can establish in humans is harder. So we're very interested in more proximate biomarkers.
Yeah.
And those biomarkers, some of them exist, some of them need to be invented. But think about these things as function. So if we can improve function, the earliest companies in longevity are going after disease of aging still. They're going after arthritis and renal failure. But soon, we're going to start seeing companies that are improving some sort of functional component, like hearing, blindness, maybe your balance, your memory. Well, if you're improving like 5 to 10 of those things, you're probably improving aging. So thinking about health span as functional longevity, that's something that people can grab onto, because so much of aging is a loss of function. Let's see if we can improve those biomarkers as a way to shortcut the development, because function is something you can measure today, whereas longevity is ultimate. And maybe improve proximate markers, we'll get the ultimate outcome we want.
So we'll come in a little bit to a possible trial that might actually be ready to get started in humans. Before I get there, I just want to float a couple of other new observations, mostly from animals, but which seem to be intriguing, and ask the panel what you think of that. One of them is this observation about senescent cells. Let me explain what that is. So your cells, which are perking along, hopefully, some of them kind of live out their lifespan, and instead of suddenly disappearing, they go into this phase called senescence, and those cells are never going to divide again, and they're eventually going to disappear. But they hang around for quite a long time. And I think we had kind of considered, I had anyway, that they were just along for the ride, and they weren't helping you, but they weren't hurting you. Turns out that may not be true. because in a mouse model, for instance, in a very clever genetic manipulation, it's been possible to figure out how to take a mouse that's achieved a certain age and has a fair number of those senescent cells and just knock them out, get rid of them right away, or keep them from ever really quite forming. And guess what? Those mice live longer. So senescent cells are not just along for the ride. They seem to be producing some negative influence on their healthy cell neighbors that's running the system down.
Right.
And there's even now some ideas about how to develop drugs that might do that without having to manipulate humans. So would any of you want to talk about that approach and how promising you see it?
I think that really gets to some real interesting, promising biology. As Francis was saying, those cells are not just sitting there quietly, taking up a little bit of space. They're spitting out pro-inflammatory, sometimes pro-even tumorigenic proteins that can go travel to other parts of you. So they're nasty characters. And the experiment in the mouse, where they genetically did a trick, unfortunately not one right now available in humans, but pointing in the right direction, those mice did really well. And so one of the things that makes a cell senescent, as we humans live our long, long lives, mice live too short for this to matter, is the telomeres. When they run down, that frayed shoelace end sends a big signal to cells and says, stop, go senescent. And so it's one of the ways cells get senescent. So, but in mice, they live too short for that to naturally happen. They go senescent for other reasons, but it's the same kind of story. So I think that's going to be a very interesting approach. One of the approaches to humans. We'll still have other things. We'll have to worry about the mitochondria, we'll have to worry about energy, have to worry about proteins getting old in some of our very long-lived cells. We have neurons that sit around for a long, long, long time, and we don't want those not functioning well. And they're not senescent cells, they're good functioning neurons. So we have different parts of us. We have to think about how to take care of the best, as Hassi was saying.
Well, let me ask Hassi about this in terms of mitochondria and energy. My postdoctoral mentor, who's a very well-known and incredibly effective and continues to be highly creative scientist now, as you can imagine, since he was my mentor, a little older than me, but he hasn't slowed down a bit. But he also restricts his calories quite severely in anticipation that that might do him some good. What is that all about?
So there's actually a caloric restriction society. There are about 3,000 people who participate in that. They eat about 70% of the calories that are recommended. They're all obviously very skinny.
You said 70, not 7, right?
70%. and they believe that they will live a long time. Now, I do want to point out that over the last 100 years, that had been the major dogma in aging research. The caloric restriction extends lifespan and health span. An interesting experiment that was published a few years ago by one of my former graduates showed that if you take your standard laboratory mice and cut their calories by 30%, they live about 50% longer. But then they took a spectrum of about 40 different genetic variants of mice and cut their calories by 30%, and about a third of them lived much longer. Another third of them lived about the same, and another third, about 10 strains of mice, actually lived less when they were calorically restricted. So just think about those guys who are caloric restricting. What if you were one of those human variants that actually lived less? You're going to be really upset.
Miserable.
And the point is that we can't just apply one-size-fits-all approach to aging. And whether there's now 20 different drugs that slightly increase lifespan in mice. And they probably will do that also in humans, but not every one of us will need the same drug. So you should be in caloric restriction, you should be in metformin, you should be in rapamycin.
I shouldn't do caloric restriction.
Nir, what should you be on?
I have a very...
Nir is going to live forever
I
no
have
matter
a
what.
very positive addition to this. You know, the least mortality in humans, men and women, across the world, across ages, is when BMI is actually between 27 and 28. That's overweight. Yep. Okay, that's me. So if you think of, you know, the experiment of caloric restriction, you already know the limits. You know that without food, they'll die fast, right? Mm-hmm. So you have to have the right number. And so the question for humans who list mortalities when they're a little bit overweight is if it has really a major role, this paradigm. Yeah. But I have to say, when it works, we want to know why it works, okay? That's the important thing. In the animals that it works, we would like to understand why it's doing it.
Like you said, we want to know the biomarkers that predict the responsiveness to caloric restriction.
But those mice, you know, the ones that responded, the ones that didn't, there's probably wonderful information in their genomes that we could, you know, start to get some real clues. I don't know if anybody's following this.
In fact,
in their
people are.
epigenomes.
Yes, absolutely.
Yeah, so we can learn a lot from that. We looked at telomeres in some people who did caloric restriction, and it actually made very little difference whether they did or didn't calorically restrict. It's only one marker. It doesn't tell you everything. But again, that was instructive. And the National Institute on Aging ran a large experiment with non-human primates and actually stopped the experiment with the taxpayers' money because the mice who were calorically restricted, even though they supplemented them very well with micronutrients, they didn't look like they were living longer. They did live some.
These were primates.
The monkeys.
The monkeys.
Non-human primates. They did better. They did have good biomarkers. Their coats looked sleek, and they did. You know, it wasn't terrible, but their lifespans
didn't live longer.
Didn't live long, and so we have to really, you know, think hard about old ideas. You know, and extract the valuable parts of the caloric restriction, but not kind of overdo the interpretation at this stage. But I think there's, you know, there's interesting things.
And I think it's really important to say that caloric restriction is such a crude approach, and we know so much more about diet today and new approaches like intermittent fasting or specific diets that limit certain macronutrients, or the Mediterranean diet, which is really the only diet that's been proven in humans to delay mortality and decrease disease.
And it's good for your telomeres.
And there's all these blue zones where people eat different things in Okinawa and in other parts of the world where people have specific diets.
Liz mentioned this, I'm sorry.
No, go ahead.
Liz mentioned this in her book, Telomeres. One of the best ways to promote your telomere health is actually to challenge it. So in a way, the best way to challenge your system is to do things a little bit differently, improve your dynamics. So maybe it's not just caloric restrict, but maybe vary it. Maybe you binge eat and then restrict yourself. You know, it's not just meditation, because our dynamic range is really narrowing, which means our homeostatic capacity is narrowing as we age and due to environmental changes. We got to expand our dynamic range as a way to extend our homeostatic capacity. So maybe it's not eating vegan, but eating vegan and then eating meat once in a while, so you don't lose that capacity. Maybe it's meditation, but then alternating with actually riding a roller coaster as a way to stretch your range. So maybe it's not one advice, but maybe it's all these different things.
Maybe it's going to a Milken conference. It sort of seems like that's a pretty good roller coaster.
And what about interval exercise?
Exercise is a kind of a stress. It's a challenge, you know, and yet it clearly has such huge salutary effects.
And
I mean, even in centenarians, I'm assuming
it has the biggest effect on health span in elderly.
In your centenarians as well.
No, I'm saying in elderly overall.
They don't need it. They don't need it.
My centenarians, they get there anyhow.
They just do it regardless.
In fact, our centenarians are 50% overweight and obese. 50%. Less than 50% are doing any exercise like walking. 60% of the men are smoking and 30% of the women. So our centenarians is not really the example of how you interact with that environment.
I'm getting a little concerned about children. Remember, they were dealt a hand of genetic cards that most of us were not, and so, you know, gene and environment, you got to optimize.
Can I quote near from something he said ages ago? It always stayed with me. You said you asked each of the centenarians, how did you think you lasted so long, so well? And you said, they gave two answers, so we all listen, right? What are the two answers? One was, I smoked, I didn't smoke, I exercised, I didn't exercise, I did this wrong, I did that wrong. And the other was, somebody in my family also lived for a very long time. In other words, genetics.
Right.
But that's the rare population that are the lucky ones, and most of us die of the things that limit our health span, right? The cancers, the diabetes, you know, the cardiovascular
Well,
Alzheimer's.
let's talk now about pharmacological interventions. So we know, again, mostly dependent on animals, much of it in mice, that the drug rapamycin reproducibly extends the lifespan of mice, and we have some idea why that works. Anybody want to explain what that is? Hassi?
So rapamycin is a fascinating molecule. It works on a pathway called mTOR, which is actually the pathway that's affected by diet and intermittent fasting. And it extends lifespan in several models, from worms all the way to larger animals. And Nir is going to talk to you about metformin in a minute, yes. But there's a fascinating large study that you wonderfully funded, thank you, that's run by a series of investigators across the country giving rapamycin to dogs. And there's already evidence that the dogs are tolerating, so it's a placebo-controlled trial, where the dog owners serve as the administrators of the drug to the dogs. The dogs are tolerating it well, and in a few years, we'll see if dogs live longer in response to rapamycin. And I feel a little bit sad that the dogs are already getting rapamycin, but Nir hasn't gotten the money, I'm sorry to say that, hasn't gotten the money yet to give metformin to people. So we have to do something about that.
So somebody once said the trouble with loving is that pets don't last long enough and people last too long. Maybe we're changing that here with rapamycin. We can equilibrate the situation. So yeah, what about metformin? Nir, you're the guy who's right out in front here in terms of not just studying that in some laboratory animal, but proposing that we might look at it in humans. What's the basis for that? What's the plan?
Right. So metformin is a drug that has existed for 60 years. It's the first drug of choice for type 2 diabetes now. It's generic, it's cheap, and it's very safe. We know all we need to do to know about metformin. But the most interesting thing from my point of view of metformin is when you give it to animals, and you can give it to nematodes and mice and rats, they would live a lot healthier and also longer. So metformin seems to be one of those drugs that is in the midst of the biology of aging. By the way, it's not far or interactive with mTOR as well. But there's other interesting thing about metformin. Since it's been here for 60 years, and since there are actually clinical trials, and when I say clinical trials, they are controlled, and also lots of observational study, we found out something interesting about metformin. Metformin, if you give to non-diabetic people, it will delay diabetes by 30%. If you give it to people with diabetes, it'll delay cardiovascular disease by 30%. People on metformin have 30% less cancer, all sources, all kinds of cancers. People with diabetes and metformin and people without diabetes on metformin have less cognitive decline. And even the most interesting of all, and this is a study that included 200,000 people out of the UK, people with diabetes and metformin have significant less mortality from non-diabetic people that are treated by the same doctors with the same risk factors. Although they are diabetic, they are more obese, and they're also more sick when they start on metformin. So if you think of everything I told you, we have already clinical evidence to suggest that what we see in animals that delay aging has already worked in humans. So we can take this data, and we planned a study that's called TAME, by the way, taming aging with metformin, or really targeting aging with metformin, and do the first proof of principle in humans to show that aging can be targeted. We expect that the effect of metformin from a very conservative assessment will be an elongation of 2 years of health span. I have another part of that, but I'll stop now.
Well, no, I think everybody will be interested then. If you were out there trying to design a trial of this sort, and you've heard the evidence that comes from the use of this drug in other situations, but it's been used to treat people with diabetes primarily. So how would you set this up? Who would be your candidates that you would seek to enroll? how would you decide whether it was working in less than 30 or 40 years? Maybe biomarkers would need to be involved. Those are the, and how would you get the regulators, we're going to come to that in a minute, to agree what would be the standard for basically accepting this as a positive result? Because it's one thing for us to say that's a good biomarker, but unless FDA agrees, then it isn't going to get you anywhere. So how have you been negotiating through all that?
Right. So this study planning needed a lot of innovation. And of course, I think you kind of understand, yeah, we would have liked to start this study in people who are 50 years old, but we'll never see the end of that, and the NIH will not pay us for such a long study. But we figured out, if we are right and what metformin is doing is targeting aging, let's take people who are older. We decided people who are 65 to 80. That's the only really inclusion criteria. At that age, they can have even any disease they need to end, because if we give them this and it will delay aging, it will delay their next disease. And this is really the second part. What are we looking for in those people? We are looking for an endpoint that is either hard endpoint, what we say. It's either cardiovascular disease or cancer or Alzheimer's or mortality. Now, we don't have to be significant in each one of them. Everyone gets a point, because aging is the risk factor for everything. And we don't know what disease you'll get first, but it's aging that's going to be the risk factor. So we have a composite of diseases, and we'll see that metformin, we'll ask whether metformin is changing the composite so that you don't get, so that with metformin, it'll take more time, if you will, to get the same amount of diseases. Now, considering that there's a lot of outcomes here, there's a lot of outcomes in people between 65 and 80, and all we need is 3,000 people in a placebo-controlled study. That is, 1,500 are getting metformin and 1,500 are getting placebo, in order to have in 4 or 5 years enough data to show that we actually delay aging.
And we need money too, to run all these things.
And what?
And we need money, we need resources.
Right.
From the investor perspective, we all want to have a lot more candidates like metformin, rapamycin out there, because we want many more shots on goal. And some of the funding comes from NIH, some of it's coming from venture. Unity Company was founded around the Senolytic idea, Senolytics, and they're screening for compounds. It'd be great to have hundreds of compounds. And the National Academy is also doing the pull side. Funding pushes, whether it's venture funding or NIH, but then the National Academy is trying to pull innovations, new technologies, through this prize mechanism that you probably all heard about. The National Academy has launched this Aging and Longevity Grand Challenge. Our family supported.
Say a bit more about that. So how would somebody actually apply and win the National Academy's Grand Challenge Prize?
Right. So the first thing that needs to happen is the prize needs to be defined. And so the National Academy, under Victor Zhao's incredible leadership and vision, has assembled a lot of the top people in the field, which includes Bob Horowitz, a Nobel laureate, Tachi Yamada, Mark Fishman. These folks are leaders of science and industry that are convening to define the criteria, and then they will solicit ideas and innovations, both from industry and from scientists, to try to hit certain targets as a way to also dramatically expand the field. And you'll be hearing more about, you can go to the National Academy of Medicine website now, and it's all defined there, although the criteria themselves will emerge through committee through a very bottoms-up process from the community, from people that are workers in the field.
So when we've done trials of interventions for Alzheimer's disease, which of course is one of those diseases of aging and one of the most feared ones, it's been really disappointing that we've been unable to see that many of the strategies that were put forward have actually provided benefit. But the argument has often been we're waiting too late in Alzheimer's disease, so that if you're already showing signs of cognitive decline, you've lost so much of your neuronal function that you're not going to bring that back. Just can't help but wonder, when you're saying now you're going to start at 65 to 80, is it possible that's too late and you'll have missed the window where metformin might have actually provided a benefit?
So that's a great question, and we made sure that we had enough data, and we went back to retrieve more data to show that there's a lot of people between 65 and 80 that were treated in metformin, and they were doing actually better than the young people on almost every aspect of that. Not only that, people who didn't get metformin because initially they didn't give people because that had kidney failure or heart attack or liver disease, but then they relaxed it, and those people are doing better. Now, you know, what a shame. They're doing better even than other people. But I want to get back to your question and to your comment. I want you to understand one thing about why I'm doing this study. I told you that I already have enough data to suggest that metformin can delay aging. The reason I'm pushing this study is very different, and that is because there's no indication that is equal to aging, okay? There's no indication. What does that mean? That means that the healthcare providers are not going to pay for a treatment that the FDA has no indication. That means that pharmaceuticals are not jumping into the field of aging because they don't see that there's money at the end for them. You know, rapamycin, which is a really effective drug, is a drug that needs more development. It's not safe in chronic treatment. In humans who are getting rapamycin, they develop diabetes, and animals develop cataract and testicular atrophy. So the drug needs a little bit more. It's a dirty drug. It needs more development and more thoughtfulness. This will not happen now. So the reason we're doing TAME is to get an indication. And the way we're doing that, we sat with the FDA, and I'll tell you, the FDA was great. Okay, because we were a bunch of scientists that are talking about the generic drug that they know everything about it. It's on you go to the web of the FDA, they know everything about metformin. We never mentioned even the word metformin. We talked about what do we have to do in a clinical study so that at the end you'll say something that's equal to aging. By the way, we don't want to say aging because aging is not a disease. Not everybody whose age has a disease, and there are ageism and all that. But we're talking about multi-composite attack on aging, multi-composite morbidity that will be a target that will be like saying aging. And this is really, for me, the great breakthrough, because we talked about all those other drugs, and this will not happen if the FDA will not pave the road for that.
And this is a transformative study, Nir. I agree completely. But I think it kind of situated in the world of medicine where cancer was 30 years ago, where you had a drug and you treated cancer. And if you think about what happened in that field in the last 30 years and how much more sophisticated we are with the concept of personalized medicine, what we call precision medicine now, we need to move into the precision longevity era, where we'll use biomarkers, mainly genetic and epigenetic, to try to predict what diseases people are going to get and give them the kind of intervention that's most likely to help them. For example, if they're prone to diabetes, we'll give them metformin. If they're prone to cancer, we'll give them rapamycin. If they're prone to Alzheimer's, perhaps we'll give them a mitochondrial peptide, and so on and so forth. And that's the future.
So, Hasi, if you were going to then advise Nir, and maybe you have, about the design of this trial, here's an opportunity to begin to get into that precision prevention of aging strategy. What should he be collecting on those 3,000 individuals that are going through this trial in terms of other measurements that might ultimately predict who responds and who doesn't? I'll ask Elizabeth the same question in a second.
DNA for genomics, DNA for epigenetic markers, plasma for proteomic markers, and for mRNA and circulating RNA markers, and then a team of statisticians who will look at that data and try to predict what is the second age-related disease that each one of them get, and whether those markers predicted that and predicted whether they got it or not. And that's going to be the future.
Liz, would you add something to those lists of all the opportunities that are here?
Yes, I think the question
And
of
telomere length, of course.
which of the biomarkers is still a really, really emerging field. You know, there's a study that came out of MD Anderson, the cancer center, where they looked at bladder cancer patients, and they said at the moment of diagnosis, did they have, of all things, depression and or short blood telomeres? Just those two combined, and sadly, immensely increased their death rate after 2.5 and 5 years. So I think the answer is going to lie in combinations of things, because we're very context dependent. I think we're not there by any means in terms of the biomarkers, because that little story just told me, wow, take two things, each of which is a weak risk factor for bladder cancer prognosis. After you equalize for everything else, combine them together, and that power became very, very predictive. So it's just the beginning. Now we get the numbers, the sophistication, the good statistics, where now you can start to combine things, because we're an integrated system. We're not just made of isolated parts. They all interact. All the biomarkers that we read out are the product of interacting parts. But it's not hopeless, because the sophistication of dealing with complex data sets, be they genomic, be they large or small marker. Depression is a very macro one. Yes. Telomere length is a very systemic one, etc. So I think we're just on the cusp of doing extraordinary things. We've got to invest in this in a very, very global way to take advantage of this potential.
So we're almost at the end, and I'm going to ask each of the panelists to give us 30 seconds of a takeaway. But before we completely go to that, I just want Nir to say, what's the status of this trial, which, by the way, we just redesigned, and I think the cost just went up about a factor of 10 because of all those biomarkers we're going to measure. And Liz wants you to measure not one thing, but two things, which means 3,000 isn't nearly enough. Anyway, what's the status of TAME?
So TAME, I hope, is going to be co-funded by the American Federation of Aging Research, and that are committed, and by the NIH and maybe several institutes of the NIH. The final review is going to be in June 16, so if that goes fine, we'll start at the end of the summer.
All right. Well, let's hear from each of the panelists a closing comment. If we could, I'll start over here this time, Hasi.
Just a shout out to personal genomics that's going to lead us to precision longevity. And I love drugs, I develop drugs, but healthy aging starts with diet and exercise.
Excellent.
2 ways you can get involved while we're waiting for the science. Get involved with the National Economy Longevity Grant Challenge. My wife and I pledged $2 million. It's a way to really support the field very broadly in all these different areas. And the second thing is for your personal health, I try to optimize my dynamic range by promoting my homeostatic capacity. Getting the homeostasis with these drugs increases atrophy. Every drug causes addiction. So do something different that actually increases your robustness.
Great.
Invest really in long-term thinking. It's going to be for the next generations as well as us. So don't just hope for quick results, because there'll be some, but there will be some real long-term benefits for our next generations.
Good point. Nir.
There is a longevity dividend that was calculated. It's really $7 trillion by the year 2050 if we can extend health span by 2 years. And the most important thing is this idea for you guys in California that maybe you'll spend 80 years to become biologically 60 is not a science fiction. It's a science that's happening now, and everybody needs to help us achieve that. Thanks.
So I hope you've gotten a good education and some inspiring words. Please join me in thanking this remarkable panel.